Hello, everybody. Nice to see you back from the coffee, and I hope you are awake from this long day. First of all, I have to thank the organizers and especially Julita to inviting me here and to be in front of you. Also, I'm impressed by the level of the conference in terms of the science and also in English level. So it's really good to hear and to be here. My topic, as you see, is about DNA electrotransfer. I will talk a little bit about electroporation, but Professor Thomas Vernier already gave introduction. It's very deep introduction and very consistent. So I will probably skip this and will go quickly. And then I will stop a little bit more on what is known about DNA delivery using electric pulses. So basically, as you know, many hydrophilic molecules, and including DNA, cannot cross easily the membrane, and usually they need carriers or vectors. And basically, most efficient are viruses that evolved along with the eukaryotic or prokaryotic cells. The cells developed defense mechanisms, but viruses also developed how to overcome these defense mechanisms. In order to facilitate and also to be safe, because working with viruses sometimes you have safety concerns, some physical methods are developed. And basically in our lab we are working with two physical methods. One is electroporation, another is sonoporation, which is ultrasound-based delivery. I will not touch this subject here, and will touch only electroporation. Basically, at some small levels, hydrophilic molecules, including DNA, can enter by endocytosis. And later on, I will tell you that electroporation seems to facilitate endocytosis. So coming back what Thomas Vernier presented already, when you put the cell in the electric field, basically you induce induced potential that can be calculated by this simple formula. But if your pulse's electric field is long enough, and you suppose that your cell is completely spherical, you can go with even more simple formula. And basically, as this formula is saying that, as Thomas mentioned, that with larger cells you develop larger induced potential. And when this potential reaches some critical value, we believe, as Thomas showed, that some pores start to develop in the membrane. But the situation is not as simple, and Thomas presented very nicely that we do not understand what's going on in the membrane. And this is just a figure for our simplicity and also for our students, saying that we have kind of permeable structures. And we believe these are pores, long-lived pores, but exactly we don't know how these pores look like. But basically what we can say that this is 3 steps: pore formation step, pore expansion step, and this pore expansion step can be, you can regard it as increase in size and number, but sometimes it can be some coalescence of the pores. And this is a very complex mixture, but still is kind of evolution of pores. And then when you switch off the electric field, these pores remains for some time, but they start to reseal. And ultimately they close up, some of them. Some of them do not close, and the cells ultimately dies. So by coming back to this formula, you can see that by increasing electric field, basically you increase the surface of the membrane you electroporate or you affect. If you maintain the same voltage, but you increase the duration of the electric field, basically what you do, you increase the defects or the pores or whatever, the structures that are increased in size. Basically, having in mind this, you can say if you want to deliver small molecules more efficiently, you can increase the voltage. But if you are working with larger molecules, probably the best choice is to increase the duration, maintaining the same voltage. So this is the message. And you can have 3 situations. Basically, if you do not reach critical voltage, so the cell does not react, and you do not revert to its permeable state. On the other hand, if the induced potential is too strong or too high, you can lead cell to irreversible electroporation. Irreversible electroporation is also, it's a difficult term. You can regard this as membrane cannot restore, or sometimes the membrane can restore, but cell still dies at later phases, at later durations, meaning that something happens inside the cell. Either it loses some metabolites, or it happens something inside. We don't know exactly what goes on inside, but we know that some cells do reseal, but they do die later on at later stages. And having this in mind, of course, sorry, I missed that one. This is most important for us. Irreversible electroporation, meaning that after you revert cell to permeable state, it remains for some time, and this allows exchange of molecules that are otherwise non-permeant. And some molecules can be delivered into the cell, and some molecules leave the cell. As Thomas showed, potassium can leave, ATP can leave, and some other molecules can leave. We do not know which molecules or which size, but definitely some molecules leave it. And this shows very nicely that you have non-permeant molecules, red dots here, and you have permeable, you deliver the pulses, you open the pores or some structures, and these non-permeant molecules enter the cell, and then the membrane closes, and you have kind of molecules delivered to the cell. In that way, you can regard electroporation as a microscalpel, that you can treat the cell membrane and deliver the molecules. And with this scalpel, you can treat many cells in one time. And this is a very good tool, a very efficient tool. And as it's shown here, there is many applications of electroporation. And most developed are tumor electrochemotherapy, gene delivery, and I will tell more about gene delivery, but there are some other applications for food processing, for bacteria inactivation, and some others. So basically I will tell you the story I did and our lab did, not chronologically, because we did in parallel. Sometimes in vivo studies we are first, and then in vitro studies. Sometimes in vitro studies we are first, and then in vivo studies. But I will try to just to show what is known in vitro, and then a little bit in vivo. Just to mention that once we do not understand how it goes in one cell, working in vivo, it's even more complex. But still, we see phenomena, and we want to understand how it goes on. So what we do, basically we prepare a cell suspension, we put between electrodes. In cell suspension you can put drugs or DNA. You deliver the pulses, and then you measure either by flow cytometry or by other means behavior of the cells. And just very quickly about electrochemotherapy, if you consider these dots as anti-cancer drug, potent in clinical, potent drug when inside of the cell. This one is bleomycin, which is a classical anti-cancer drug which is used in clinics, but it's not very efficient because it cannot cross easily the membrane. But if you help this with electroporation, then it enters easily and it kills the cell because it acts as endonuclease. And this were our first experiments after Louis Mir developed that technique. We checked in our lab, and this is bleomycin concentration, and this is cell viability. And you have a curve of viability curve without electric pulses, and you have viability curve with electric pulses. And you can see that you can decrease bleomycin concentration basically 1,000 times and have the same efficiency of cell killing. This is very, very efficient. And of course, Lewis Muir had this in mind to combine bleomycin and electric fields. And if you can do this in vitro, you can do this in vivo. And our experiments, very old experiments, almost 20 years ago we did this. And the first experiments we were obtaining that if you have a control, tumor grows, they grow along during the time, and you use bleomycin or electric pulses separately, tumors develop in the same rate. But if you combine both, you first inject drug and then deliver pulses to the tumor, then you see very strong effect, anti-tumor effect. And later on it was developed in tumors. You can see the efficiency, and then it was developed in clinics. It was developed technique, electrodes, and it was used for tumor treatment. And as you can see, it's very, very efficient. 6 months after the treatment, tumor is gone. So it was in many places in Europe, especially, not in States so far, as I know, but it's going to be. And it's also can be used in veterinary to treat animals. And I stop here with electrochemotherapy and start with gene delivery. So basically, the first study that showed that electroporation can be used to transfer DNA into the cells was from, we say, a father of electroporation, Eberhard Neumann. I just knew that he was born in Wroclaw. This is something new for me. Actually, what he did, he used the pulses, 8 kilowatts per centimeter, 5 microsecond, exponentially decaying pulses. And he delivered thymidine kinase into the cells. And he showed for the first time that indeed you can deliver the gene. And he showed a window from 6 to 10 kilovolts, up to 6 kilowatts, it happens nothing. After 10 kilovolts, you probably start killing the cells, and they are killed not they're killed by pulses themselves. And very interesting is that he proposed a model how it can happen. He was thinking that maybe DNA absorbs at the surface of the cell, and he imagined that maybe magnesium as a divalent ion can contribute to this, and he increased concentration of the magnesium chloride, but he saw that some magnesium is needed, but with higher concentration of magnesium, you decrease the efficiency of delivery. So basically, the first proposition of the first model was that DNA somehow absorbs at the membrane. Then you have poration, and DNA just slides across the pore inside the cell, and then you have transfection. Of course, DNA has to enter the nucleus in order to be transcribed and translated. So right now, with development of the reporter genes, it's become more easy to follow gene transfection. So this is plasmid coding GFP, gene fluorescent protein. So you have a bright light image, and then you have fluorescent image. So basically what you did, you just put the plasmid in the cells, in monolayer of the cells. You put the electrodes, you deliver the pulses, and you see some cells fluorescent, meaning that some cells uptook DNA, and this DNA was transcribed and translated. The main difference between small molecule delivery is that DNA, if added after the pulse, does not enter the cells. So meaning that DNA has to be before the pulses in order to be transferred. With small molecules, you can deliver the pulses and put the small molecules after the pulse, and still molecules will enter the cell. With DNA, this does not work. So DNA has to be present during the pulse. And this, of course, gives some clues what's going on. But there's one problem. Thomas mentioned that we do not know about the size of the pores. But according to some preliminary estimations, according to diffusion of small molecules inside the porated cell, we can estimate that the range of the pores is from a little bit below 1 nanometer up to 10 nanometers. But if you consider the plasmid, and it's a big molecule, and it can easily exceed 100 nanometers. So basically you have a problem how to put a big molecule through the very narrow pore, as an elephant through the small doors. And actually the first scientists who started to think about how it happens, they started to imagine. And the first studies were that coming from Tismajev group, it's Sukharev and Tismajev. They started to think maybe DNA is, of course, it's polyanionic molecule that's very sensitive to electrophoresis. And in an electric field, it just moves along the electric field. And if you have a pore, the current is concentrated in the pore, and DNA can be pushed through the pore, and the membrane just becomes larger. And basically they did that. They put some dextrans that were not able to enter the cell without DNA. But if they added DNA and delivered the pulses and then put dextrans, those dextrans that were not able to enter without DNA entered with DNA. So basically it seems that this model is more, it has some basis, some background. Or it can be some endocytotic pathways. It's not exactly endocytotic-like pathways. They're proposing not considering endocytosis itself. There are some other mechanisms, how they proposed to the same group, that DNA can be elongated in some way, not as a shred, but elongated in the electric field, and it can cross in that way, or as a shred. If you consider linear DNA, not plasmid, or plasmid as well, it can insert at one end into the pore, and then is somehow uptaken by the cell. Or it can interact with many of the pores, and then can be pushed across the membrane. Some other models propose that maybe you have some very huge pores for a short time, and during these huge pores, maybe DNA can cross at the moment. Some other models propose that there are probably interaction with the membrane. There are specific lipids that are positively charged, and as DNA is negatively charged, maybe there is interaction of the DNA and the membrane lipids. And during this interaction, when we deliver the pulses, there is some forces that act on the DNA and it enters the cell. Still some other models proposed, it's more or less like previously showed, that DNA at some point interacts with DNA and then crosses it, or it really interacts with DNA, making some membrane DNA complexes, and in that way it goes in. But everything started to be more clear when Tessier and Roll's group and a special lady, Golgio, showed that after delivery of the pulses, you have some patches on the membrane. Basically what they did, they delivered the pulses in the presence of DNA, and then they put the TOTO dye, which labels DNA. And what they saw that on one surface, you have some small patches at the cathodic site. And these patches remain stable for some time. they form in millisecond range, but then they stay for some time. And later on, minutes after that, these patches, fluorescent patches, enter the cell. Not immediately like small molecules, but they enter the cells in the range of the minutes. And in the range of hours, the DNA gets expressed. And if you compare that with the small molecules, extraction or delivery, you create the pores, and immediately after that, the diffusion across the membrane starts, and it takes for some time. So, of course, you can see here that the mechanism of DNA delivered into the cell is completely different from the small molecule diffusion. They quantified the structures, the size of these structures, and the size of these structures was something in the range of 100 to 500 nanometers. And they proposed that these structures, very huge structures, probably could enter the cell by endocytosis. But also they showed that if you deliver not one pulse, but second pulse, third pulse, fourth pulse, what you do, you increase the fluorescence of the patches. But what they say, that the number of the patches does not change. It stays the same, just you increase the fluorescence of the patches. What does mean? Probably that DNA, the fluorescent DNA, goes in the same place and somehow forms a large aggregate. How this happens, they do not explain, and we do not understand. Considering, if you consider that you want to get the largest expression, what you can do, you can play with the electric pulses, or another parameter what you can do, you can increase the concentration of the plasmid. And we just did the same study, just wanted to see dependence of the transfection efficiency on concentration of the plasmid. And what we did here, it's from 100 micrograms to 600 micrograms. And indeed, we increased up to 60% of cells that were transfected. But what we saw also that with increase of the plasmid concentration, you start to decrease the viability of the cells. And we do not know, and we do not understand what's going on, how the plasmid kills huge plasmid concentrations, how they kills the cells. But according to preliminary studies, what we found in literature, there are two possible ways. One way is that when we put too much of the DNA inside the cell, some defense mechanisms of the cell starts to fight against the foreign DNA. And it can be two possible pathways, maybe some other pathways, but one is formation of inflammatosome, and also in endosome there's some tool-like receptors that react with these endocytotic pathways and can induce apoptosis or some other death pathways.
Could you maybe more towards
Sorry. Okay. So then the another approach how to analyze how DNA enters the cells was to really look at the endocytotic pathways. So the group from Rolls started to see what happens at the site of the membrane when you deliver the pulses in the presence of DNA. Well, they saw that there is formation of the actin threads, actin filaments, at the level of the membrane. So this is also the sign of endocytosis. And because of that, and because of other hints, they started to really look at the endocytotic pathways. So they used many endocytosis inhibitors. As you know, there are many endocytotic pathways. And these inhibitors inhibit different endocytotic pathways. And here is the control when you deliver the pulse and you have some cells transfected, it gives you 100%. And then when you use these endostotic inhibitors, you decrease transfection efficiency, and this decrease is usually concentration dependent. So they showed that many available endostosis inhibitors could decrease transfection efficiency. And also what they did, they tried to see whether they labeled DNA, and also they had transfected cells that express some markers in endosomes. And they looked that there is they merged the image, and they saw that labeled DNA corresponds to these endosomes. And this was kind of proof that DNA stays in endosomes. And of course, this proves also that this is kind of endostotic pathways. They quantified different endostotic pathways, and what they show here that there are some, not one endostotic pathways, but different endostotic pathways taking place during the same mechanism, during the same transfection procedure. What also they did, they looked at the pathways in the cytoplasm, how DNA moves in the cytoplasm. They did some correlation curves, looking at the labeled plasmid movement and also endosome movement, and they see again correlation. So all these hints show that probably, or already for sure, that the DNA when interacts with the membrane then enters into the cell by endostotic pathways. And so they did the graph. So as you see here, delivery of the pulses do permeabilization, and then DNA can interact with the permeabilized membrane. There is formation of actin network, and then there are different endostotic pathways that allows DNA enter into the cell, and they travel into the cell, and there's another obstacle that DNA, in order to be expressed, has to overcome. This is enter the nucleus. And this is still don't know, not yet explained in detail how this happens. So basically right now I switch from in vitro studies to in vivo. I did a postdoc in Louis Meyer lab, the guy who developed electrochemotherapy, and later on he switched on gene delivery, and he invited me to work with him for more than 1 year. Basically, it was already developed technique that you can use DNA injecting into the muscle, expose the muscle to the electric pulses, and you can have expression in the muscle. But what Louis proposed to me work on is to use a different combination of pulses. Basically, people before used either these pulses or these pulses. This is high voltage short duration pulses, and this is lower voltage but a little bit longer duration pulses. And Louis had the idea that maybe you can use high voltage short pulse, I will call it HV pulse, and then low voltage long duration pulses, I call them LV pulses, in order to see whether you need to electroporate, what is the importance of the electrophoresis in the muscle or in delivery of gene, how it plays in efficiency. So what we did here, we just use these standard pulses. Standard pulses, I mean these pulses, because they were shown to be most efficient for muscle transfection. And what we did here, we just injected DNA and delivered the pulses some minutes after DNA injection into the muscle. And what we managed to show that DNA stays for at least 4 hours without any degradation. And you can inject DNA into the muscle and go for the lunch. And then coming back, you can deliver the pulse to the muscle. And you have quite a high transfection. So basically, DNA stays in the muscle for 4 hours, non-degraded. Also, what was very interesting, we used a radioactive marker to follow up on the muscle permeabilization. And what we did here, we permeabilized the muscle and then injected at different time points this radioactive marker. And as you can see here, there's no pulse. Okay, it uptakes a little bit radioactive marker, but 3,000 seconds after, which is about 1 hour, you inject radioactive marker and you still have some delivery into the cell. Meaning that it's very unexpected result for us that after muscle permeabilization stays for a long time, almost 1 hour. And actually, this allowed us to develop a very clever experiment. What we did here, if you just deliver DNA alone, you have very low transfection. You deliver DNA and then HV pulse, this pulse one, so we have some transfection. If you deliver LV pulse, just like this one, so we have lower transfection. Then if you inject DNA and then you deliver HV pulse, wait for 100 seconds, deliver LV pulse, then you have increase in transfection efficiency. But as we saw that permeabilization stays for a long time, and DNA can stay in the muscle non-degraded for a very long time. So what we did, basically we pulsed the muscle, injected DNA, we had a time lag for 100 seconds. During that time we injected DNA and then we pulsed with LV pulse. And basically what we saw, it's exactly the same. Basically, what we did, we dissociated HV pulse from the LV pulse. And what we showed that the muscle needs to be permeabilized, and then with using LV pulses do not permeabilize, just do exerts electrophoretic forces, and LV just brings DNA across the membrane, or increase efficiency at least. So if you do in reverse, LV, DNA, 100 seconds, HV, there's no transfection, or almost no transfection. So with this, we actually proved that for efficient delivery of DNA, LV pulses plays important role. And then we wanted to really see the importance of HV pulse and LV pulses. And what we did here, we maintained LV pulses stable and just played with the HV pulses. We tried to optimize HV pulses. We increased either duration or number of pulses, and basically we got exactly the same transfection efficiency. So HV has to be, but once the muscle fibers are permeabilized, then you have gene delivery. Then we played with the LV pulses. We used exactly the same HV pulse, but optimized LV pulses. We increased the number, duration, and we saw that independence of LV pulses, DNA transfection efficiency changes a lot. With increase of the duration or number of pulses, you increase the transfection efficiency. And actually, how we measured transfection efficiency, basically we used the luciferase gene. We lysed the muscle and measured luminescence. And also we did with GFP. And if you look at the muscle fibers, you see that a lot of muscle fibers are transfected. But basically when we cross-sectioned these muscles, we saw that not so many muscle fibers are transfected. And actually, this was a big deal for us. Then back in Lithuania, we tried to really have close to 100% of transfected fibers, but we never succeeded to have them. Basically what we succeeded something like 10 to 20%. And basically we tried to optimize in many ways. One way was to inject more rapidly the solution in the muscle fibers, helping to distribute the solution of the DNA inside the muscles. Or we use some enzymes that destroy contacts between the muscle fibers. But still, we do not succeed to overcome 20%. So then we come back in vitro studies, trying to understand in more detail whether these HV and LV pulses can be optimized in vitro studies. What we did here, we just used these pulses, and we tried to optimize the gap between HV and LV pulses. We used one HV, one LV pulse, and we just tried to shorten the duration between HV and LV pulses. And we saw here that with 1 microsecond, I just skip a little bit, that here it's more evident than when you use 1 microsecond gap, basically you increase the transfection efficiency. What we also showed that if you increase viscosity of the medium and you deliver the pulses, this is the same viscosity as here, but here we delivered bleomycin with the HV and LV pulses, and we saw that LV pulses for delivery of bleomycin are not so important as for DNA. For DNA, LV pulses here is HV pulse, and light gray is HV plus LV pulse. And we saw that the high viscosity, the more efficient LV pulses were in our studies. So basically from in vitro, in vivo studies, we came to the same conclusions that other groups came, that DNA delivery is needed. In order to deliver DNA, first you need to permeabilize the cells. Then you control the efficiency of the delivery by controlling the electrophoretic component of the electric pulses. Here we already know more, that there is some endostotic pathways taking place, and here we do not understand too much how DNA enters the nucleus. What I can say that although we do not understand all mechanisms how DNA enters the cells, but already some clinical trials using electrogene therapy undergoing. And also my last few slides is some very exotic, very difficult technique, which is called in utero electroporation. I was working for some time with developmental neuroscience, and people in that field use also electroporation. And what they do, they want to follow the axons pathway and how the guidance molecules control the growth of the axons. And in order to do that, they can do this in vitro studies, but most interesting is to do in vivo. And what they do, they have a pregnant mouse. They basically operate the pregnant mouse. They take out the uterus, but not the embryos from uterus. They use a glass pipette to inject a micro solution of the DNA, and then they put the electrodes, as it's shown here, and they deliver the pulses. And they showed that then they put the embryos back to the mother, and allow to mother to deliver the babies a few days later. And what they showed that according to the position of the electrodes, you can deliver DNA in the specific areas of the brain, either cortex or the hippocampus or some other places. And they showed that you can definitely have a very high transfection efficiency, and this allows you to follow up your studies in vivo. If you just inhibit or express some specific receptors, then you can see how the axon pathway, axon growth is disturbed or modified. The pulses they used, but we are quite low, 50 volts, using 4 to 5 millimeters electrodes. And also, what they showed that if you use 2 positive electrodes on the side and the negative electrodes, negative electrodes on the side and positive electrodes on the top, you also can control where you can deliver the gene. Here, with one pulse, they delivered the gene in 2 hemispheres. So with this, I just conclude. I have to acknowledge my students, my PhD students, master students, and of course many collaborations that we have in our lab. And thank you for your attention.
Thank you very much for the excellent lecture. Do you have any questions? One question?
Okay, very happy that I have it here.
Thank you for this fantastic lecture. We were wondering actually if you ever measured the influence of the temperature of the stability of pores, let's say, during electrotransfer. Is it possible, for example, to keep cells from resealing by temperature and therefore maybe enable the electrotransfer and then increase the temperature?
Basically, what people show in electroporation field, that with decrease of the temperature, the duration of the lifetime of pores increases. But of course, when you decrease the temperature, the diffusion of the molecules also decrease. And I don't know the studies, maybe Thomas know, how the delivery is dependent on the temperature. Maybe Thomas can comment. I don't know exactly that one.
Question.
You mentioned the clinical trials with the gene transfections, which could be used as a cure.
Yeah.
And this first phase was quite many years ago.
Yeah.
Has been used in wider applications, or it stopped for some reason, and what is the problem with that? We don't hear about this.
It stopped for the reason that it's not so effective. It helps to cure, it helps to treat, but it does not cure. And what people are looking for immuno combinations, for immunotherapy, and this study, the first study, was with interleukin-2, and since it showed some efficiency, but was not very efficient, so some additional progress is needed.
Because I remember the first studies and it was very exciting, and then it's like I never heard about this anymore.
The same with gene therapy in general. With viral vectors, the same. It was at some point was exclamation. It's going to be cured all the diseases, but then some deaths occurred, and the gene therapy just stopped, and then it's coming back again.
Thank you.
Do you have any more questions? If no, thank you very much. I would like to close this part of the lectures.
Okay, thank you.
And now this is the most difficult part of our meeting, the awards. It was really difficult to select the best presentations because all of you presented high top level of science, and we really appreciate that you